Well, first of all, on behalf of Professor Scott O'Neill, the leader of the Eliminate Dengue program and based in Monash, and myself, thank you for the invitation from the committee to come and attend this meeting. Whilst we just get the PowerPoint started, just by way of background, the project Eliminate Dengue that I hope to give you an update on is a little bit different from what we've been talking about previously at the meeting today. The aim of the project is to use a naturally occurring intracellular bacteria called Wolbachia to try and reduce the ability of Aedes aegypti to transmit dengue viruses. So the aim is to not eliminate or reduce the population of mosquitoes, so that's a normal control method to try and stop dengue, but to actually reduce the ability of the mosquitoes themselves to transmit. I've just got a short video I'd like to show you that perhaps summarises some of the background around the project. We can just get the video started. The screen seems to be flicking a bit. Is there any way that we can try and address that? Maybe we can just run with this the way it is. Okay, next slide please. So this video I'd like to show you is a thing called speed draw, where someone, just don't start it yet please Petrina, where someone can draw or illustrate a concept and then someone videotapes that and then we can speed that up and use that as a story to explain some of the background. I'd like to perhaps use that today to give you an overview of how we want to use Wolbachia, and then perhaps follow up with some of the research we've been doing and some of the field trials we've been doing in Australia and Vietnam. Thanks, Petrina. So the target for our project is Aedes aegypti. Aedes aegypti originated out of Africa several hundred years ago and then spread around the world, as Scott explained earlier. We think on sailing ships, on water barrels. It's now present in tropical and subtropical areas throughout the world. It's responsible for the transmission of dengue and in some areas yellow fever. Our project aims to use a naturally occurring intracellular bacteria called Wolbachia. So it lives inside insect cells and is transmitted from the female to her offspring, and in the case of mosquitoes, through her eggs. So it's not infectious. Wolbachia is very common in the environment. We estimate that somewhere around 60 to 70% of insects naturally contain Wolbachia, including insects here in Yogyakarta that bite people already, including Aedes albopictus and Culex quinquefasciatus, so insects and mosquitoes that are biting people commonly in the environment. The reason why Wolbachia is so common is that it can manipulate or change the way insects reproduce. You can imagine a female insect without Wolbachia. If she mates with a male insect with Wolbachia, she can lay eggs in the environment, but none of those eggs hatch. So effectively the Wolbachia is sterilising the female insect if she doesn't have Wolbachia. You can imagine a female insect, if she has Wolbachia, she can mate with an uninfected male, all of her eggs hatch, and all of her offspring have Wolbachia. And similarly, if both of your parents have Wolbachia and you're an insect, all of their offspring are normal, they have Wolbachia, and they all hatch. And they all survive, both males and females. So this is occurring around us as we speak and is allowing Wolbachia to establish itself in insect populations around the world. This happens naturally. So over a few generations, Wolbachia can go from a low percentage in the insect population to close to 100%. We were particularly interested at the beginning of the project in a strain of Wolbachia called the popcorn strain, and it was found to shorten the lifespan of insects in the laboratory, in particular a fruit fly species. We know that mosquitoes, a very old mosquito in the laboratory, could be 30 days of age. And we know the transmission of dengue requires a period of replication inside the female mosquito before that mosquito can transmit. So essentially it's the old or geriatric mosquitoes in the population are the most dangerous in terms of transmission of dengue. So we had an idea. What if we could move Wolbachia from the fruit fly that shortens the lifespan of the adults into the mosquito? Could we shorten the lifespan of the mosquito before she's old enough to transmit? And then we thought if we could establish these mosquitoes in the field, what would be the impact on transmission of dengue? So that was this challenge we had about 7 or 8 years ago. We knew moving Wolbachia from the fruit fly into Aedes aegypti, so it's not naturally occurring in Aedes aegypti, was going to be very challenging. To do that, we needed to try to inject newly laid mosquito eggs using a very fine needle with purified Wolbachia. So this took tens of years to do, thousands of attempts, and then in 2009 we were successful. We had a colony of mosquitoes that contained Wolbachia, and the females passed it on to her offspring. When we looked at how long those mosquitoes live in the laboratory, we found, like the fruit fly, they had a 50% reduced lifespan. So this was very positive. What was unexpected, and this is one of the moments in science that comes along very rarely, is that we found when we tried to feed these mosquitoes dengue virus, the Wolbachia was blocking the virus from replicating inside the mosquito. So it didn't matter how long the mosquitoes live. If the mosquito can't become infected with dengue to start off with, it can't transmit later on. So this was unexpected, very positive. So we wanted to move from the laboratory into the field. So we built a large field cage in Cairns in northern Australia. And into that cage we put wild-type Aedes aegypti mosquitoes from Cairns without Wolbachia in them. Then each week we released a small number of mosquitoes that contained Wolbachia, and we monitored that cage over time to see if Wolbachia could establish in the mosquito population. After 3 months, close to 100% of the mosquitoes in that cage had Wolbachia. So this is a proof that we could maybe implement this in the field. Now I'd like to tell you about some field trials we've been doing where we've actually released mosquitoes with Wolbachia into its natural habitat. And that's around people's houses. So in Cairns we've released mosquitoes with Wolbachia. They mate with the wild mosquitoes in the population. The Wolbachia transfers onto that wild mosquito population, and all the mosquitoes in those areas now contain Wolbachia and have a reduced ability to transmit dengue. So our goal going forward is to try and utilise Wolbachia as a tool to try and reduce dengue. So it exists in insects throughout the world already. We're trying to move it into something like Aedes aegypti and use it to reduce transmission of viruses like dengue, and hopefully make an impact on this number. So this is the number of dengue infections we estimate occur every year. So our goal is in the future to try and reduce that number down. Next slide, please. So perhaps just to highlight some of the science around the project a bit more. So just a very quick update on Wolbachia itself. So it's very common in insects, and it's not infectious. So it can't be transmitted in a mosquito bite from an insect to a human. It only lives inside insect cells, so it's an intracellular bacteria. And in mosquitoes can only be transmitted from the female through her eggs to the next generation. Interestingly, Aedes aegypti has never been found to be naturally infected with Wolbachia, nor any of the Anopheles species that are involved in transmission of malaria. We're not quite sure why. Next slide, please. The safety of this is paramount in our mind. And so we've done a lot of experiments over the last 10 years to show that Wolbachia cannot transfer from mosquitoes to any animal when it feeds. In fact, with 60% of insects naturally infected with Wolbachia, no mammal or vertebrate animal has ever been found to be infected with Wolbachia. We commonly eat Wolbachia. You may not realise that Wolbachia is an insect that contaminate our food supplies, and we're eating remnants of Wolbachia regularly. And we've also had volunteers in Australia and Vietnam, even here in Yogyakarta, that have been feeding mosquitoes in the laboratory that contain Wolbachia for many years. And we've been able to screen their blood and monitor their blood going forward, and no one's ever had any antibodies or any evidence of any side effects of exposing themselves to Wolbachia mosquitoes. Next slide. It's just a short video showing how difficult it was to actually transfer Wolbachia in. So Wolbachia doesn't move very easily from one species to another. These are newly laid Aedes aegypti eggs within 1 hour of being laid, before the shell of the mosquito egg becomes hard. And you can see the injection of purified Wolbachia into the eggs. So this Wolbachia was collected from fruit flies and then purified in the laboratory and then re-injected back into the eggs. So the Wolbachia is not modified. It's not a genetically modified organism. It simply moved from one insect into Aedes aegypti. This is done tens of thousands of times, and you may end up with 1 or 2 female mosquitoes that have Wolbachia. And then you can keep those mosquitoes that survive in a cage in the laboratory and start a colony of Wolbachia mosquitoes, and that's essentially what we've done. Next slide. So this moment in science when you see these unexpected results, and in our case we thought they were very positive, and these are some results looking at the replication of dengue virus in mosquitoes that have Wolbachia. These 2 different types of Wolbachia that are in Aedes aegypti mosquitoes and the wild type mosquitoes from Cairns, from northern Australia. And the Wolbachia is reducing the replication of dengue virus inside these mosquitoes. And these experiments have been repeated many times. And so simply having Wolbachia present in the mosquito is interfering with the virus replication inside the insect. Next slide. These experiments that we've moved on to now in collaboration with colleagues at Cam Simmons' group and Katie's group at Oxford University and Ho Chi Minh City. And they've a lab close to a large referral hospital for tropical disease. So hundreds of patients can come in a single week with acute dengue infection. We're able to take a sample of their blood. So these are patients that have high dengue virus viremias. And then we can put that blood into a glass feeder that we can expose mosquitoes to in the laboratory. We have mosquitoes with Wolbachia and the wild type mosquitoes, and we can compare how dengue is replicating, and we can test the saliva of the mosquitoes and see whether those mosquitoes are able to transmit later on. Next slide. These are some results, and so it's a little complicated, the graph, but I'd just like to walk you through it. So this is a dengue 2 virus that was being fed to Wolbachia infected and uninfected mosquitoes. And the patient had quite a high viremia, 9.1 logs. And these are the results of the mosquitoes after 10 days. So the mosquitoes were fed on the virus, then held in the laboratory alive for 10 days, and then we process the mosquitoes and test them for virus. And the blue is wild-type mosquitoes, actually here from Jogjakarta. The red is a strain of mosquitoes that has a W-MEL Wolbachia, again from Jogjakarta, and a W-MEL pop, so 2 different types of Wolbachia. The most important panel here, and the tissues that are probably most relevant, or the samples are most relevant, is the presence of virus in the saliva we collect from the mosquitoes. So after 10 days, the wild type mosquitoes from Jogja, about 25% had virus present in the saliva, but the Wolbachia mosquitoes were negative. Next slide. Similarly, at 14 days, we had 70% of the natural Jogja mosquitoes without Wolbachia were capable of transmitting, or at least have virus present in their saliva. And similarly, none in the Wolbachia mosquitoes. Next slide. And similarly, even mosquitoes that are very old, 18 days after they were fed, we weren't able to detect virus. So this was a very positive result and quite a strong challenge because these are natural viruses that are circulating. Next slide. So to summarise, close to maybe 300 different patients that we've taken blood from and exposed to mosquitoes, there's 2 different trends. And these are really different Wolbachia strains that are having a different effect on the virus. So effectively, wild-type mosquitoes, you can feed them virus, then after a certain time period, most of them will transmit. The effect of the popcorn strain is it blocks transmission. No matter how long those mosquitoes live, they're not capable of transmitting. The W-MEL strain has a more subtle effect. It reduces the infection in the mosquitoes and delays the time period. So both of these effects, if we hand this data to the modelers, they predict if we could establish this mosquito across a geographic area, dengue transmission would collapse. It wouldn't be able to be sustained. This one here, under most transmission cycles, you'd be looking at a reduction of dengue transmission below R0, which means you're not going to get large epidemics. And so approximately this, under current epidemiological situations, could provide a sustainable solution to reducing dengue transmission over a large area. Next slide. And this effect of blocking pathogens is quite broad. So it doesn't just apply to dengue, it also applies to the range of other pathogens, including yellow fever, chikungunya, and a range of filarial nematodes, and recently was shown to be introduced into anopheline species and block the reproduction of Plasmodium. So this effect of virus blocking, pathogen blocking, seems to be quite broad. Next slide. So this is just a section through mosquitoes that have different types of Wolbachia in them. So this is the one that I'll tell you about soon for releases in cans, and the green shows the tissues where Wolbachia is located. So many insects, many mosquitoes have Wolbachia in naturally. The most important consideration is where in the mosquito the Wolbachia lies. So we know that dengue virus replicates in the midgut and the fat bodies and the salivary glands of the mosquito. We find that mosquitoes have the Wolbachia in those tissues are the ones that interferes with dengue replication. So we're testing these strains of Wolbachia that can reduce dengue transmission. Next slide, please. So compared to a conventional control program using insecticides or some other method, the aim is to reduce the population of mosquitoes. Our approach is to reduce the ability of the mosquitoes to transmit the virus, not the population of mosquitoes itself. To do this, we need to release into local areas mosquitoes that have Wolbachia. This is the only way of introducing Wolbachia into field populations of mosquitoes. And we do that by undertaking weekly releases of mosquitoes with Wolbachia. We get to a certain stage where we can stop releasing, and then Wolbachia will drive into the remaining mosquito population in that area and remain in the mosquito population thereafter. Next slide, please. So a critical component is we need to have a critical threshold or frequency of Wolbachia in the mosquitoes in a local area before Wolbachia will establish or drive into that population. Simply releasing 1 or 2 mosquitoes into a local area won't be sufficient to get across this 30% approximately threshold. So the aim of these releases is to have the frequency in the mosquitoes above this point, and then we can stop releasing and the Wolbachia should establish. And that's what we've been testing. Next slide. The trials we've been undertaken in Australia has commenced in 2011. But before that, we started a conversation with communities about Wolbachia and how it could potentially be used. You can imagine if I came and knocked on your door as a householder and said, I'd like to release some mosquitoes at your house with a bacteria inside them. They're going to feed on you, your children, your neighbours, and they're going to lay all their eggs back in the environment. And we hope that this will stop dengue. Would you be happy for us to do that? I'd probably say no, I don't really want you to do that. So we were aware that this science is quite new and novel, and people may be a bit uncertain about it. So before the release, we undertook 2 or 3 years worth of engagement, a conversation with these communities to explain about Wolbachia. We then obtained approval from the Australian government to undertake the releases, and then also did a risk assessment to show that this wasn't going to present any unique risk to the Australian environment. Next slide. And then we're particularly careful about explaining this to communities, sharing the results with them so that they become involved in the research themselves. Next slide. We chose 2 communities in Northern Australia, and these are approximately 1,000 houses in each community. Located, this is the centre of Cairns, a large city, and we chose 2 areas that were north and south of the city, and they were small communities and isolated. And by isolated, they were surrounded by habitat that was not suitable for Aedes aegypti. So these first releases were undertaken in areas where we minimised the risk of the mosquitoes and the Wolbachia spreading outside of those areas. Next slide. And we obtained consent or approval from each house to release mosquitoes at each house. Next slide. And each of these green dots represents the location of those houses where people said, we're okay for you to release mosquitoes in and around our house. And the red dots represented people that said, I'd prefer not to be involved in the release. So you can do the release, but I don't want you to release mosquitoes around my house. We said, that's okay. Next slide. And there's 4 components to the release. Rearing the mosquitoes, releasing them, monitor them using various mosquito traps, and then testing the mosquitoes in the laboratory to determine which mosquitoes that we collected and what proportion of them have Wolbachia. Next slide, please. And we communicated all these results back to the communities, and we published these results every month in local newspapers to share the results with them. Next slide. So these are the first trials. So we undertook 9 and 10 weeks of Wolbachia releases in 2 communities in 2011. After 9 and 10 weeks, the frequency of Wolbachia in the local mosquito populations was around above 60%. And we said, we believe we can stop releasing mosquitoes now. But we continued to monitor, so every month we went back and collected mosquitoes and tested them for Wolbachia. Within 12 months, essentially 100% of mosquitoes in both of those areas contained Wolbachia and had a reduced ability to transmit dengue. We've monitored this for close to 3 years now. It'll be 3 years in January. And over that time period, we see some fluctuations during the dry season when mosquito numbers are very low. But essentially, 100% of mosquitoes in both of those areas have Wolbachia. And we bring those mosquitoes back into the lab and feed them dengue virus. They still have the same level of virus blocking. Next slide. So at the stage now, earlier this year, we're looking at different methods of trying to deploy or use this technology in the field in Australia. And so initially we released adult mosquitoes into the field. Now we're deploying or using other methods, such as plastic buckets that we can put some water in and put some Wolbachia mosquito eggs in and put those at people's houses, and the mosquitoes can emerge. So using both of these methods, and we've got 2 additional communities, and we've used both methods in each area. Next slide. Shows that you can adapt the release method to suit the local situation. And both of these communities now, we believe Wolbachia will become established, like those first 2 communities. So the technology can be adapted to suit the local situation. Next slide. We'll skip over these. So most of the trials we've done so far have been taking mosquitoes from the laboratory that contain Wolbachia and releasing them into small areas, around 1,000 houses. After a series of releases, we stopped releasing and we continued to monitor. We want to learn how can we maybe deploy this on a bigger scale. If we had a city of maybe 400,000 people, how could we do this a bit more efficiently? Next slide. So you can imagine a city that's made up of a series of blocks or houses. Can we release mosquitoes from the laboratory into these smaller areas, and will mosquitoes spread into nearby areas and carry Wolbachia with it? That's what we aim to test. And so we'd like to be able to deploy on a small scale, but on a large city, and hopefully Wolbachia will then protect all the mosquitoes in that city from virus. Next slide. So we're currently doing this in Cairns. At the beginning of the year, we selected 3 different areas within Cairns City itself. So these areas are not isolated like the first sites, but you can see here there's all houses and businesses through there. But we selected 3 different size areas: a large one, approximately 1,500 houses, a medium-sized one, and a small one. So we released mosquitoes each week into each of those areas. And we wanted to test whether Wolbachia can spread into neighbouring areas and protect the nearby mosquitoes. Next slide. So this is some of the raw data. So each of these spots represents one of those mosquito traps. And the size of the dot represents the number of mosquitoes we collected that week from that trap. And the green proportion represents what proportion of those mosquitoes have Wolbachia. So this inner red box is the area that we release mosquitoes into. We never released any mosquitoes outside here, but we monitor outside the area. So now we're seeing in nearby areas, this is 100 metres away, we're starting to see 60% of mosquitoes have Wolbachia. In 200 metres, about 12% of mosquitoes. So Wolbachia is starting to spread into the nearby areas because there's no barriers to mosquito movement here. Next slide. And so across all 3 areas we're seeing establishment. The smaller area may indicate that that release area was too small for Wolbachia to establish. So we're learning what is the critical way to deploy the technology across a larger city. Next slide. So whilst most of this work has been done in Australia, and that choice was made consciously, that we want to develop the technology and test it in Australia first before asking other countries to consider using it. But now we've got collaborations across 7 different sites, including here in Jakarta, but also in Vietnam and Brazil. Next slide. So in Vietnam, they undertook a release earlier this year on a small island in central Vietnam. And instead of our staff or their project staff releasing the mosquitoes, they utilised the local community members. There's 40 community members they asked to volunteer, and the community members undertook the releases of the mosquitoes themselves. Next slide. These are some of the communication materials the project there came up with to communicate to the local people about where the mosquitoes come from. So they came from the Ministry of Health lab in Hanoi. They flew on a plane to the local city of Nha Trang. Then they went on a boat across the local island. And then the local community collected them. And they put a small container. Next slide, please. And just follow on. Next slide. And they put a small container inside each house with the mosquito pupae, so the aquatic stage, and these mosquito pupae contained Wolbachia. And the mosquitoes emerge into the houses. So this was testing it in a different way. And these are some of the volunteers that worked for 26 weeks on the project. Thank you. Next slide. So whilst in Australia we released the WML strain, and was shown to be successful, in Vietnam they selected the very strong virus-blocking strain, the popcorn strain. Whilst they got the Wolbachia frequency quite high, so it approached 90% at the end of the release, when they stopped releasing, the Wolbachia frequency started to decline. Probably indicates that this type of Wolbachia is not ideally suited to the local environment in Vietnam. So they plan on continuing to monitor the release, and then if it drops off to close to zero, they're proposing to do a second release using the WML strain, the one that was shown to be successful in Australia. Next slide. So I won't talk anymore about the project specifically here in Yogyakarta, except to say that over 2 years ago we started a collaboration with the Faculty of Medicine here, University Gadjah Mada, and we're pleased to get funding from Yayasan Tahir, a foundation here in Indonesia that funded work previously around trying to prevent dengue here in Yogyakarta. So they were keen or excited about looking at an alternative approach to try and trial here in Yogyakarta. And I'll leave Wasito to explain more about that in the next talk. Next slide. We have a collaboration with the Ministry of Health and its research institutes in Brazil. And so we've been working with Luciano Moreira, one of the key scientists in Brazil, for many years. And so Luciano said to us, do you realise that Brazil spends $1 billion a year trying to control dengue without much success? So hopefully in May next year, the city of Rio de Janeiro will undertake small releases of Wolbachia mosquitoes in some of those key areas. If that research seems to be positive, they hope to move forward with larger trials later on. Next slide. So this is some of the project team, and we see this as a research collaboration across many different countries. An important part of that collaboration is bringing people together to share experiences from different projects. And we're fortunate enough to travel to Bali earlier this year and share some of those experiences. Next slide. So this talk has really summarised work over the last 10 years, and over 200 different people now are contributing to this project globally. And these are some of the key people and institutions that made significant contributions over those years, so I'd like to acknowledge them. And also thank you for your attention, and thank you to the committee again for the invitation to come and present. Thank you.
Thank you.